22.5 beneficial prokaryotes. By the end of this section, you will be able to do the following. Explain the need for nitrogen fixation and how it is accomplished. Describe the beneficial effects of bacteria that colonize our skin and digestive tracts. Identify prokaryotes used during the processing of food. Describe the use of prokaryotes in bioremediation. Fortunately, only a few species of prokaryotes are pathogenic. Prokaryotes also interact with humans and other organisms in a number of ways that are beneficial. For example, prokaryotes are major participants in the carbon and nitrogen cycles. They produce or process nutrients in the digestive tracts of humans and other animals. Prokaryotes are used in the production of some human foods, and also have been recruited for the degradation of hazardous materials. In fact, our life would not be possible without prokaryotes. Cooperation between bacteria and eukaryotes. Nitrogen fixation. Nitrogen is a very important element to living things, because it is part of nucleotides and amino acids that are the building blocks of nucleic acids and proteins, respectively. Nitrogen is usually the most limiting element in terrestrial ecosystems, with atmospheric nitrogen, N2, providing the largest pool of available nitrogen. However, eukaryotes cannot use atmospheric, gaseous nitrogen to synthesize macromolecules. Fortunately, nitrogen can be fixed, meaning it is converted into a more accessible form, ammonia, NH3, either biologically or abiotically. Abiotic nitrogen fixation occurs as a result of physical processes such as lightning or by industrial processes. Biological nitrogen fixation, BNF, is exclusively carried out by prokaryotes, soil bacteria, cyanobacteria, and Frankia spp. Filamentous bacteria interacting with actinorhizal plants such as alder, bayberry, and sweet fern. After photosynthesis, BNF is the most important biological process on Earth. The overall nitrogen fixation equation below represents a series of redox reactions. Pi stands for inorganic phosphate. The total fixed nitrogen through BNF is about 100 to 180 million metric tons per year, which contributes about 65% of the nitrogen used in agriculture. Cyanobacteria are the most important nitrogen fixers in aquatic environments. In soil, members of the genera Clostridium and Azotobacter are examples of free-living, nitrogen-fixing bacteria. Other bacteria live symbiotically with legume plants, providing the most important source of fixed nitrogen. Symbionts may fix more nitrogen in soils than free-living organisms by a factor of 10. Soil bacteria, collectively called rhizobia, are able to symbiotically interact with legumes to form nodules, specialized structures where nitrogen fixation occurs. Figure 22.27. Nitrogenase, the enzyme that fixes nitrogen, is inactivated by oxygen, so the nodule provides an oxygen-free area for nitrogen fixation to take place. The oxygen is sequestered by a form of 562. Chapter 22. Prokaryotes. Bacteria and archaea. Access for free at openstax.org. Plant hemoglobin called leghemoglobin, which protects the nitrogenase, but releases enough oxygen to support respiratory activity. Symbiotic nitrogen fixation provides a natural and inexpensive plant fertilizer. It reduces atmospheric nitrogen to ammonia, which is easily usable by plants. The use of legumes is an excellent alternative to chemical fertilization and is of special interest to sustainable agriculture, which seeks to minimize the use of chemicals and conserve natural resources. Through symbiotic nitrogen fixation, the plant benefits from using an endless source of nitrogen, the atmosphere. The bacteria benefit from using photosynthates, carbohydrates produced during photosynthesis, from the plant and having a protected niche. In addition, the soil benefits from being naturally fertilized. Therefore, the use of rhizobia as biofertilizers is a sustainable practice. Why are legumes so important? Some, like soybeans, are key sources of agricultural protein. Some of the most important legumes consumed by humans are soybeans, peanuts, peas, chickpeas, and beans. Other legumes, such as alfalfa, are used to feed cattle. Figure 22.27. Nitrogen fixation nodules on soybean roots. Soybean, Glycine max, is a legume that interacts symbiotically with the soil bacterium Bradyrhizobium japonicum to form specialized structures on the roots called nodules where nitrogen fixation occurs. Credit: USDA, everyday connection microbes on the human body. The commensal bacteria that inhabit our skin and gastrointestinal tract do a host of good things for us. They protect us from pathogens, help us digest our food, and produce some of our vitamins and other nutrients. These activities have been known for a long time. More recently, scientists have gathered evidence that these bacteria may also help regulate our moods, influence our activity levels, and even help control weight by affecting our food choices and absorption patterns. The Human Microbiome Project has begun the process of cataloging our normal bacteria and archaea, so we can better understand these functions. A particularly fascinating example of our normal flora relates to our digestive systems. People who take high doses of antibiotics tend to lose many of their normal gut bacteria, allowing a naturally antibiotic-resistant species called Clostridium difficile to overgrow and cause severe gastric problems, especially chronic diarrhea, figure 22.28. Obviously, trying to treat this problem with antibiotics only makes it worse. However, it has been successfully treated by giving the patients fecal transplants from healthy donors to re-establish the normal intestinal microbial community. Clinical trials are underway to ensure the safety and effectiveness of this technique. 22.5. Beneficial. Prokaryotes. 563 Early biotechnology. Cheese, bread, wine, beer, and yogurt. According to the United Nations Convention on Biological Diversity, biotechnology is any technological application that uses biological systems, living organisms, or derivatives thereof, to make or modify products or processes for specific use. 5. The concept of specific use involves some sort of commercial application. Genetic engineering, artificial selection, antibiotic production, and cell culture are current topics of study in biotechnology and will be described in later chapters. However, humans were using prokaryotes before the term biotechnology was even coined. Some of the products of this early biotechnology are as familiar as cheese, bread, wine, beer, and yogurt, which employ both bacteria and other microbes, such as yeast, a fungus, figure 22.29. Figure 22.28 Clostridium difficile. This scanning electron micrograph shows Clostridium difficile, a gram-positive, rod-shaped bacterium that causes severe diarrhea. Infection commonly occurs after the normal gut fauna are eradicated by antibiotics, and in the hospital can be deadly to seriously ill patients. Credit. Modification of work by CDC, HHS. Scale bar data from Matt Russell. Scientists are also discovering that the absence of certain key microbes from our intestinal tract may set us up for a variety of problems. This seems to be particularly true regarding the appropriate functioning of the immune system. There are intriguing findings that suggest that the absence of these microbes is an important contributor to the development of allergies and some autoimmune disorders. Research is currently underway to test whether adding certain microbes to our internal ecosystem may help in the treatment of these problems, as well as in treating some forms of autism. 5. http://www.cbd.int/convention/articles/question_a=cbd-02 http://openstax.org/l/un_convention United Nations Convention on Biological Diversity. Article 2. Use of terms. 564 Chapter 22. Prokaryotes. Bacteria and archaea. Access for free at openstax.org. Figure 22.29 Some foods produced by microorganisms. Some of the products derived from the use of prokaryotes in early biotechnology include a. Cheese, b. Wine, c. Beer and bread, and d. Yogurt. Credit bread. Modification of work by F. Rodrigo, Wikimedia Commons. Credit wine. Modification of work by John Sullivan. Credit beer and bread. Modification of work by Chris Miller. Credit yogurt. Modification of work by John Sullivan. Cheese production began around 4,000 to 7,000 years ago when humans began to breed animals and process their milk. Fermentation in this case preserves nutrients. Milk will spoil relatively quickly, but when processed as cheese, it is more stable. As for beer, the oldest records of brewing are about 6,000 years old and were an integral part of the Sumerian culture. Evidence indicates that the Sumerians discovered fermentation by chance. Wine has been produced for about 4,500 years, and evidence suggests that cultured milk products, like yogurt, have existed for at least 4,000 years. Using prokaryotes to clean up our planet. Bioremediation. Microbial bioremediation is the use of prokaryotes, or microbial metabolism, to remove pollutants. Bioremediation has been used to remove agricultural chemicals, e.g., pesticides, fertilizers, that leach from soil into groundwater and the subsurface. Certain toxic metals and oxides, such as selenium and arsenic compounds, can also be removed from water by bioremediation. The reduction of CO4-2 to CO3-2 and to SaO, metallic selenium, is a method used to remove selenium ions from water. Mercury, Hg, is an example of a toxic metal that can be removed from an environment by bioremediation. As an active ingredient of some pesticides, mercury is used in industry and is also a byproduct of certain processes, such as battery production. Methyl mercury is usually present in very low concentrations in natural environments, but it is highly toxic because it accumulates in living tissues. Several species of bacteria can carry out the biotransformation of toxic mercury into non-toxic forms. These bacteria, such as Pseudomonas aeruginosa, can convert Hg+2 into Hg0, which is non-toxic to humans. One of the most useful and interesting examples of the use of prokaryotes for bioremediation purposes is the cleanup of oil spills. The significance of prokaryotes to petroleum bioremediation has been demonstrated in several oil spills in recent years, such as the Exxon Valdez spill in Alaska, 1989, Figure 22.30, the Prestige oil spill in Spain, 2002, the spill into the Mediterranean from a Lebanon power plant, 2006, and more recently, the BP oil spill in the Gulf of Mexico, 2010. In the case of oil spills in the ocean, ongoing natural bioremediation tends to occur, since there are oil-consuming bacteria in the ocean prior to the spill. In addition to these naturally occurring oil-degrading bacteria, humans select and engineer bacteria that possess the same capability with increased efficacy and spectrum of hydrocarbon compounds that can be processed. Bioremediation is 22.5. Beneficial prokaryotes 565 enhanced by the addition of inorganic nutrients that help bacteria to grow. Some hydrocarbon-degrading bacteria feed on hydrocarbons in the oil droplet, breaking down the hydrocarbons into smaller subunits. Some species, such as Alcanivorax borcumensis, produce surfactants that solubilize the oil, making it soluble in water, whereas other bacteria degrade the oil into carbon dioxide. Under ideal conditions, it has been reported that up to 80% of the non-volatile components in oil can be degraded within 1 year of the spill. Other oil fractions containing aromatic and highly branched hydrocarbon chains are more difficult to remove and remain in the environment for longer periods of time. Figure 22.30 Prokaryotes and bioremediation. A. Cleaning up oil. After the Exxon Valdez spill in Alaska, workers hosed oil from beaches and then used a floating boom to corral the oil, which was finally skimmed from the water surface. Some species of bacteria are able to solubilize and degrade the oil. B. One of the most catastrophic consequences of oil spills is the damage to fauna. Credit A. Modification of work by NOAA. Credit B. Modification of work by GOLUBENKOV. Go. Saving Taman.